I'm going to tell you a little bit about our organization. Again, I'm Joanne Geer, Executive Director of the Biopharma Research Council. We essentially are an association for scientists through all the different silos in biomedical research. So that's industry, academia, nonprofit, government, and supplier labs and their teams. This morning you're going to be hearing about global collaboration to address Ebola from Dr. Howard Young from the NCI and Dan Didier from Thermo Fisher Scientific. I'll be introducing them in just a moment. I just want to share with you a few of our other activities. Just last week we had an introduction to the microbiome, which is online now. You can see those recordings. And in the spring we'll be doing a short course in the microbiome on these dates in April, and you'll be learning more about that. And we are also planning a physical conference in the Bethesda area after that. In March, we'll be holding a wonderful executive retreat on best practices in outsourcing, preclinical and early clinical. This will bring together folks from the pharmaceutical biotech side in terms of those who are purchasing services from outsourced sources, and then CROs and other organizations that help them. This summer we'll be going back to North Carolina to Research Triangle Park. This will be actually our fourth research symposium. It's a terrific one-day program where folks across the region share their work, and it's a really wonderful interactions on that particular day. And next fall we'll be doing our second version of D2D, Data to Drugs and Diagnostics. Our organization is really about helping to build various cohorts of communication amongst researchers and the services that they need, and obviously the connection between researchers and their generation, collection, analysis, sharing, and security of data is very key. So that's what this conference is all about. You can ask questions through the question box at any time, and we will handle all the questions after both of our presenters have spoken, but feel free to do that. So I will now introduce our speakers. First, we're going to hear from Howard Young. Dr. Howard Young received his PhD from the Department of Microbiology and Immunology at the University of Washington and did postdoctoral research under Drs. Edward Skolnick and Wade Parks at the National Cancer Institute. He joined the NCI in 1983 and became Deputy Chief of the Laboratory of Experimental Immunology in 2006. Most recently, his laboratory has developed novel mouse models of lupus and aplastic anemia based on chronic expression of low levels of this important immunoregulatory molecule in the murine C57BL6 genetic background and the BALBC genetic backgrounds, respectively. He is also collaborating with a laboratory in Malaysia in the development of probiotic microorganisms as carriers for vaccine delivery systems. Dr. Dan Didier brings more than 30 years of experience to his role as head of public health at Thermo Fisher Scientific. He works closely with the U.S. CDC, PHE, WHO, and their affiliates around the world. He provides congressional briefings and White House debriefings. Before joining Applied Biosystems, which later became Life Technologies and subsequently Thermo Fisher Scientific, Dan was a serial entrepreneur. He was founder, president, and CEO of Cytogenomics, SilicoCyte, NewGene, DNA Tree, Script Alert, and Healthy Biome. Prior to starting these companies, he was president of Insight Genomics Asia and a research head at Monsanto. And next we will hear from Dr. Young.
Thank you, Joanne. I think this this topic is particularly appropriate the day after Time Magazine announced that their People of the Year are the five individuals who are on the front line of treating Ebola in Africa, and I think that is a recognition that is most appropriate, and these are really heroes because they went into a situation of no hope and brought hope to the people there and have helped save lives. So to give the audience a little bit of background on me, I'm a member of the Cancer and Inflammation Program, which is based on the Fort Detrick campus in Frederick, Maryland. And because of our location, about 10 years ago, I began to interact with United States Army scientists who work for the Research Institute on Infectious Diseases to study the consequences of Ebola virus infection on gene expression in monocytes and macrophages. And in fact, I was fortunate enough to be involved in what was really an amazing study done about 10 years ago on a serial sacrifice of 18 non-human primates that were infected by Ebola Zaire, where 3 animals were sacrificed each day for 6 consecutive days in order to understand the pathogenesis of Ebola virus infection. And I think that study couldn't have been done anywhere else in the world except at Fort Detrick at that time. And even today they're going back to the tissue blocks to help understand what happened there in order to provide a better understanding of what's happening in the people who become infected with the virus. The slide I'm showing everybody is in fact publications on Ebola, and I think it's very interesting. The first publication was in 1977, and this is all based on PubMed data, and you can see that the number of publications was relatively small. And I think in the early days, the research on Ebola was led by 2 individuals in the U.S., C.J. Peters, who was at the CDC, and Peter Jarling, who for much of his career was at the Army and their Infectious Disease Research Institute. Peter then worked with Nancy Jacks and mentored Tom Geisbert and Lisa Hensley, who have become leading Ebola research professionals on their own. And for many of the papers we see in the first 10 to 15 years have 1 or more of those authors on the papers. And I think the explosion not only is part of the trend in biomedical research to go where the money is, but the ability to study individual Ebola genes has now increased with their availability, and so people are using specific genes to look at how Ebola can combat the interferon response, to look at how Ebola replicates, and to develop new tools and therapeutics for blocking the virus at many different avenues of the infectious cycle. I think it's one of the things that I've always been interested in is the interferon response, and it's not that Ebola doesn't invoke an interferon response. In fact, in the non-human primate study, there were high levels of circulating alpha interferon, but there's only a weak beta interferon initial response, and I think this may reflect upon the inability of the host immune system to control the infection. And I think one of the major questions today is why are some people surviving infection, and I think that will lead to better understanding of how the host is responding and can manage to stave off the infection in some individuals, while in the majority of individuals they cannot unless they're carefully treated with both new agents that are coming on board as well as maintaining their fluid balances and receiving the best care possible in light of no specific approved drugs yet for this infection. But I think one of the things challenging us is how to do some of this basic research. To give you one example, I've asked 4 of the frontline Ebola investigators, people who are actually doing virus work in BL4 conditions, if you treat human monocytes with interferon gamma, which is the gene I've been studying for a long time, are they resistant to Ebola virus infection? And no one had the answer to that. One of the individuals said, well, we know if we do that with HeLa cells, we can show resistance. But it might be something as simple as giving a low dose of gamma interferon, which has already been approved for human use, could help increase the protection of individuals before they go into the hospital settings or the frontline settings. But all of these, there are many ideas that people have about research on the virus, and I think one of the gaps right now is how can some of these ideas get actually tested. I know the U.S. Army has a website where they welcome new ideas about not only Ebola but other infections that may impact on troops around the world, and the NIAID has also had set up a site where people could go and present ideas for potential consideration. So we're in an era of obviously increasing publications, as you can see from the slide, and I think that number will probably double or triple within the next couple of years as people get access to the reagents and can work with individual Ebola virus genes without needing BL3 or specifically BL4 conditions. And so with that, I'll turn it over to Dr. Didier to talk to us about his thoughts about Ebola virus.
Thanks, Howard. I think everyone should be able to see my slides now. And one thing I want to do is, thank the organizers for allowing me to speak, but also, like Howard said, I'd actually like to applaud Time for choosing the Ebola fighters as the Person of the Year. So often the CDC, the Public Health Labs, the World Health Organization, the Doctors Without Borders, the people that are frontline don't get the credit they deserve. They're putting themselves in harm's way in many cases, and it's a courageous effort that they do. And so I just want to say thank you and applaud you for what you do, and applaud Time for recognizing them, because they do not get the recognition that they deserve. What I'm going to do today is I'm going to first give a little background on Thermo Fisher Scientific, a little background on Ebola, and then the ways that Thermo Scientific collaborates in the prevention, the diagnosis, and then the safety equipment that they provide to these labs and to the people out there that are doing either Ebola research or actually on the front lines with Ebola. Thermo Fisher Scientific, it's a growing company. We have over 50,000 employees. We clearly are a world leader in serving science. There's 10,000 people on the field out there. Many of you are probably familiar with our premium brands, our Life Technology, our Applied Biosystems, our Invitrogen brands, our Thermo Scientific brands, our Fisher Scientific, and our Unity Lab services. So we're out there. We're in every lab around the globe. If you're not using our products, you're probably not in science, because we are around the globe in many respects. Our mission is to enable our customers to make the world healthier and cleaner. We don't do it. We're the ones that work with you. You're the ones that make it healthier, cleaner, and safer. We rely on you to do this. We provide you the tools and hopefully collaborate with you to make these things happen and allow your science to go forward. If you look at emerging and pandemic infections and threats, there's many out there, many that we've seen over the years that have come through. You're seeing more and more these days because, last few weeks was traveling around Asia. I was in 7 countries in 14 days. If I had some disease, I could have spread it all over, not only to those people on the many planes and hours of air travel I was on, but also many of the meetings I had. So things very rapidly move out there. Ebola, if we contain it, we don't see it move, but it's still an issue. It's still out there. It's still a threat to us. Pretty much going to read this slide because I think it's so important. The Ebola outbreak in Western Africa has really reached epidemic levels. Over 7,500 people have been affected and 6,300 have died. There's still experimental treatments with vaccines are underway and treatments, but they're still in very early ages, very early stages. Again, that increased global travel, it's what's causing these to spread. And especially when you have something that doesn't show symptoms for as much as 21 days, you can be infected and be infecting other people potentially when you get to those designations. Our mission, Thermo Fisher Scientific's mission to enable our customers to make the world healthier, cleaner, and safer, feeds very well into collaborative nature combating this dangerous disease. Our technologies and collaborative efforts are involved in prevention, diagnosis, and safety equipment. The current Ebola outbreak is the largest outbreak, and it's growing exponentially with a doubling period of every almost 35 days. It's genetic similarity between the species. They've sequenced the 2004 strain. Is a natural reservoir. It's followed human-to-human transmission during this outbreak. The confirmed cases have already been more than the total historic outbreak case numbers. So how does Thermo Fisher work in collaboration with people? we support the quest for the prevention of the disease. We collaborate with research searching for vaccines. We offer media and equipment to grow and store these vaccines if one should be supplied. Our bioproduction for vaccine production, those that are done in cell-based cultures, are essential. We work with these groups to ensure that the media mix is optimized for their vaccine production so that they can get the highest yields possible as well as the quickest yields possible. In Ebola monitoring and containment, we work closely with the U.S. CDC, the Department of Defense, and others in their efforts to contain the further spread of the Ebola virus. Some of the technologies that we have that are used in these cases. Sanger sequencing is still the gold standard in sequencing. It's been used for many years. Now it's, capillary-based systems. For those companies that are producing vaccines, want to ensure they have the correct strain before they spend tens of millions of dollars on media to produce these vaccines. They ensure that the sequence of the strain that they're using is correct. Sanger sequencing is used in these cases. Our ION AmpliSeq panel, this is our next generation sequencing panel. It's a very rapid method, and I'll go into more details on it as well as Qpcr, but it's a very rapid method of getting sequence from these strains that are out there and circulating. We can track mutations, can use it in research settings. It's used in research settings in public health labs, CDC, and other areas around. Qpcr. Qpcr, again, it's an established method. We know it works. I'll talk a little more about it in a couple more slides, but we know that it works. We know that it's accurate. We know that it's sensitive. It's used for the confirmation of Ebola patients with the symptoms. We can leverage the current Qpcr infrastructure that's out there, the systems that are in place around the globe, and these are the systems that have been approved for emergency use authorization as well as just studies in the research market setting. Everyone's, I'm sure, familiar with the Qpcr workflow. You collect the sample. Again, the sample needs to be collected in such a way that you do not transmit virus from one person to another person. RNA is isolated in the case of the Ebola virus. We do a one-step RT and then a real-time PCR to read it. Using this system, collaborating with the DoD, CDC, they received emergency use authorization so they can very rapidly test and diagnose the disease. The worst thing you can do is have someone come into a hospital setting that has malaria or dengue, and they have a high fever. You don't know if it's Ebola or not. You isolate them with another Ebola patient, and they end up getting Ebola. This way you can very rapidly diagnose that they either have Ebola or they do not have Ebola. I'm going to shift directions now to the ion Torrent semiconductor sequencing, next generation sequencing method. Again, what it does is it copies the DNA, it's loaded on a chip, nucleic acids are incorporated, and it reads it in a very rapid mechanism. What we've done is we actually have produced an AmpliSeq panel. So what an AmpliSeq panel does is it targets that specific virus, in this case Ebola. So you can take a blood sample from one of these individuals, you isolate the RNA, you then sequence the sample. In our case, we have it such that it sequences Ebola only, as well as some control human for positive controls to ensure the system is working. This way you can check and see if there are reassortments, rearrangements, mutations that are occurring in the Ebola strains that are out there. It's very rapid in its mechanism. It enriches, again, that DNA or RNA, in this case the RNA from the Ebola virus, so that from that single sample you can just look at that. And this is what our collaborators are doing out there. This is why we designed it with them in mind, working with them to very rapidly pull it out there, not have the background of all the other human DNA that's in there. And then you can analyze it, and within 2 days you can have the analysis done and see if there are changes occurring within the Ebola virus. Safety equipment. Thermo Fisher Scientific is a large organization. We distribute many products that are being used out there. Howard and I were talking before the seminar began about the spacesuits that are put out there. You know, those are the type of things. The hoods that are put out there in the BSL-3, BSL-4 laboratories that are used. Preventing people from getting the disease using that safety equipment. It's only through our global collaborations and global collaborations of others that we'll beat this Ebola outbreak in Western Africa. Tens of thousands of people's lives depend on it. Prevention, experimental treatments, vaccinations, and detective systems are required. Collaborative efforts are required to make this happen. I like this last timeline. It refers back to the initial timeline I showed you earlier. We're discussing Ebola today, but what is it going to be tomorrow? We know in Madagascar they had the bubonic plague outbreak recently. Other outbreaks are going to occur. We know that the World Health Organization, the global health community still needs a lot of work. We need to strengthen that. We need to invest in that. We need to make sure that it is strong so we can either contain or we can prevent or we can treat these diseases as they come out because it's a threat to our global environment. With that, I want to thank you for your time, and I think we're going to go to questions next.
Dan, I'd like to start off by asking a question. What are you doing to maximize the amount of material that you can get from like a single serum sample? For example, there might be that people want to see if the infected individuals have expressed antibodies to Ebola to determine what epitopes. I'm curious, for example, on what interferon alpha subtypes might be involved in the response of people who survive, because there are 13 different interferon alpha genes, including beta interferon. Is there a difference in their response because the different interferon genes have different effects on virus replication? Other people may want to look at RNA from PBMC and so forth. So what are the best mechanisms, or how do you see maximizing the limited sample availability?
You know, that's a great question because limited sample availability is so important here. Even for us to try to get samples to test our system, because we produced our own Ebola system. So that's always the difficulty here of getting those samples. You know, as we discussed earlier, it's a small amount of blood that's taken one time from one of these patients. I don't know the answer to your question. Maybe that's something for the larger group here on the phone, is how do we do that? How do we, you know, preserve as well as disseminate these samples so more people can have access to them, so more research can be done on them? That's a great question. You know, I know there's a number of manufacturers of kits that don't have access to any of these samples because, again, you know, they have to be in a containment system, which most laboratories don't have.
I'd like to ask a question. Could you speak a bit, Dr. Didier, about the organizations or universities and companies that you're collaborating with? And obviously you're creating new technology or testing new technology in new ways, but I'd like to hear about the other pain points that you're hearing. Sampling seems key. How does the state of healthcare on the ground affect the ability for researchers to make progress and so on?
Yeah, you know, so access to samples, right, which I mentioned, that is always an issue. We work closely with the CDC. They do a tremendous job. They're not applauded enough for what they do. We need to ensure they get plenty of money and infrastructure, as well as the NIH, and you know, this is the strength in the U.S. anyways. But there's actually global efforts that are required here to make this happen and to get those things in place. We work with Public Health England. We work with the Ivory Coast labs. We get in front of, all of them. We do train the trainers in some of those situations where there's someone on the field that can actually be there and know how to do it. CDC has, I think, 200 people over there doing testing, putting themselves in front of these people and working with them. World Health Organization is also, highly involved. They definitely don't have enough money going into their efforts on a global basis. My travels around Asia and everywhere, these people, this is a disease, especially Ebola, that can, spread very rapidly if it's not contained. And so the concerns are there of someone coming in. So we have to work very closely with them to ensure that they have the system. It's not only us. I mean, CDC works very closely with these. The public health labs work very closely with these and their counterparts around the globe.
We have a question.
I'd like to add that investigators here, I first want to just expand on that investigators from here in Frederick, from the Army and the Navy, are on the front lines. They're setting up the hospitals in West Africa, and they're also involved in the direct testing of the blood samples. So it truly is a worldwide effort to help the West African countries combat this disease.
Yeah, and Howard, thanks for pointing that out. I didn't mean to step over the Department of Defense. It's just I'm not used to them being so closely involved, and they really are here, and the work they're doing is tremendous in putting those hospitals in place, and as well as the infrastructure that's required, even the power supply. Some of these regions don't have power for 8 hours, and so in order to even to do these tests and even to treat these people. So yeah, the work that's done there abroad as well as here, the samples that come in, and the work that you've done as well.
Thank you. That ties nicely to our question, which I'll read in its entirety. So not all of Africa has electricity. Does Thermo Fisher have a non-electronic-based assay to flag possible onset of infectious diseases?
And that's a great question. We're always looking at new technologies and new ways to do it now with smartphones. I don't know why they have a phone part in there. Maybe you could, to call it a tricorder someday. Those are the type of things we need to push more and more for. What we've done in this case is we work with some collaborators to actually put mobile detection systems in place that actually contain power supplies there as well. The CDC, I know, has a number of those as well in place, where they have, I think, Honda generators on there that are driving these instruments. But yeah, the smaller and smaller we can get, those point-of-care type of rapid devices, we still have to remember, because there's a number of ideas coming out there, they still have to be accurate and sensitive. If it's not accurate and you got a false positive or false negative, or they're not sensitive where you think that the person actually doesn't have it and does have it. So we have to, there's a lot of concerns that have to be done here, but the question is an excellent one, and hopefully one of these days we'll have a solution for that.
Here's another from our audience. This is from Tom Fair, who is our Director for Strategic Alliances here at the BRC. Has a consortium of companies been formed to detect Ebola? If so, what solutions might be forthcoming? So focusing on the company relationships.
Yeah, so I know we work with a number of companies and a number of different technologies and look at those things. I know other companies do as well. It's not, Thermo Fisher Scientific only that does this, but we do look at a number of companies and ways we can work. Some just from the point of view that they have a good technology and we have a global distribution network to put that in place. Others where we can use our scientific clout and their scientific clout to make this happen. A true consortium where people have gone out and looked at it, I don't know of, many other than, a few worth of 2 or 3 together, but not a larger amount. On the vaccine side, we do look. We don't actually produce vaccines ourselves. We work with the vaccine manufacturers, but again, it's not exactly what you're asking for there.
Well, I appreciate things have been moving so quickly that it may be a while before such a consortium can form. But
well, there is a WHO advisory panel, at least on potential treatments for Ebola, and a couple of my close colleagues are on that panel. But I have to admit, I don't know if it includes companies for diagnosis. But the WHO has brought together experts from around the world to consider different approaches.
Yeah. And we do that with all, Ebola, with other viruses as well, and that's important to get that scientific brain trust to see how we can combat these things. So.
Let's talk a bit about software to track the spread of the disease and support epidemiological studies. So I appreciate, as a public health person, I'm sure that there are lots of different platforms that you're working with. Is there any that are specifically focused on Ebola or that include Ebola in a panel?
So Howard, are you guys looking at that at all? I know there is a lot that the CDC does and the World Health Organization. We follow actually their lead here and don't actually have our own software.
It's being done at the Broad Institute, so a lot of work at the Broad is following Ebola epidemiology based in Boston, and I think they've developed some software packages to do that.
Great. What are the speaker's thoughts on monitoring the immune system for early detection of Ebola infection versus trying to detect Ebola directly?
Howard, that one's yours.
So that was part of our serial study in the non-human primates, and I actually was charged with looking at cytokine and chemokine expression in the blood during the infection, and as early as 3 days we could see the increase. And so certainly I would argue that you could look for an interferon response early, but that, of course, one has to keep that in mind, that many of these patients also have other diseases and they can trigger an interferon response. So I think part of the role, and I know this is going to be done at the Broad as well, is that they want to look at the genes expressed very early in the patients, given the availability of the samples, to try to look to see, okay, what are the biomarkers that could indicate that this is an Ebola virus infection. But as I said, these patients have malaria, some patients have HIV, so it's going to be, you need a lot of data to weed it out and define those specific genes. But I think it's quite possible that a subset of interferon-inducible genes might be recognized as being associated with Ebola virus infection, and you could have an early panel to look for the coordinate induction of such genes that would be very valuable. Because I think the key is understanding the early immune response to virus infection, so we need to look at that in many different ways.
Yeah, and I think, with the technologies these days, especially with next generation sequencing, either to look at the whole genome response or to look at specific targeted panels of genes, you can do that very rapidly and at a cost that's not prohibitive. So those are some areas that, we could make great strides.
Great. Let's see. What efforts have been made to develop a cold chain storage link from infected regions to places that could ship samples? Is that still being done by FedEx? Is that basically how the samples are reaching the U.S.?
Yeah. So the shipment of samples is a tricky area, and they have to have specific guidelines for that. I don't know if FedEx is the method or not. I know that I saw from a leader at FedEx what their guidelines were, and these are potentially dangerous, and there are shipping containers, and there's whole protocols that are written for these shipping of samples, as well as who can receive these samples. The key here is with a live virus or not, as well as even samples that have been inactivated. There's questions of shipping there as well because of the safety involved with it and who receives these things. I don't know, and I'm sure there are. It's outside my privy of people that are looking at this. But there are protocols in place for that.
We're doing some terrific questions here. Have groups started pre-screening populations in the path of infection to get a baseline prior to infection? That is, look for changes rather than absolute responses as an early flag.
I'm unaware that that's being done. It's a really terrific idea, but it would require a close coordination with the health agencies of the various governments, and also earning the trust of the people. I think many of the audience may know that there has been a problem with getting proper treatment because there's been a distrust of those coming in and thinking that they are bringing the virus with them, and they're not there to help the population. So the governments involved have to build trust with their populations in order to get baseline samples. But again, that's going to be complicated by the many other diseases that many of these people already have. But it would be a useful idea. And I also wanted to take a moment to direct everyone to the work of Pardis Sabeti, that's S-A-B-E-T-I, at the Broad. She's really the leader in the epidemiology of Ebola virus infection. And I just heard her talk a week ago here at Fort Detrick, and you should follow her work because it's really answering questions about how the virus changes and how they can track the viruses among the population based on changes in the viral sequence.
Yeah, thank you for mentioning her. In fact, it was a wonderful program that Howard's group hosted, and I got a chance to chat with her briefly, and she has agreed in principle to do a webinar for us. So hopefully in early 2015 we'll be able to hear directly from her. We're going to just launch a very, very short poll. I'm going to launch it right now. It'll just take a moment. If you could click on the best answer to this question, whether your organization is currently working on Ebola. Great, 5 more seconds. All right, so I don't know if everybody can see this, but 75% say they're not working on Ebola yet but are thinking about it. 13% are just getting started, and 13% have been at it for a while. For those of you who are, there we go, I can share that. For those of you who have been at it for a while, if you'll make a note in the question box, we can actually give you the floor for a moment, and you can talk about what's going on in your lab if you like. I'm going to ask the next question, but if you'd like to share your thoughts, we'd be happy to do it. The next question here is, do healthcare workers get tested before entering an infected area and after working in the region? The question is whether the concern about returning healthcare workers can be managed.
So each healthcare area has their own protocols for looking at that, and whether they're tested or they're isolated. We've seen a lot of that. It's a political issue in some regards here, because they go in harm's way, and then we want to isolate them for 21 days. And then the other question there is, the testing. All the tests are done when they're symptomatic. We, can probably pick it up before they're symptomatic, depending on the viral titer and when symptoms occur. Extensive studies haven't been done there. They really should be done there because we know our kit can detect on the 25 viruses, and so we should be able to pick it up before symptoms occur. But they are routinely screened for fever, and if they do show symptoms, but as well as that's monitored closely. But it's different protocols for each group.
But obviously, treatment of people if they have evidence of infection but before they're asymptomatic would be much more successful, and that would be an ultimate goal because you want to get them early in the infection, you want to protect their immune system, boost it if possible, and they would be much more likely, I think, to survive disease if you could do that.
Yeah, the earlier better in all disease cases, as well as, we get vaccines out there. A number of groups are working on vaccines. We get those vaccines approved out there and used. That would be very helpful.
For the cases that we have seen that have come to the U.S., it seems that there hasn't been additional infection. If I'm remembering correctly, we haven't managed to save every patient, but we've managed to not then infect the healthcare workers that are taking care of them. And first of all, is that your understanding as well? Am I remembering that clearly? I'm not remembering a case.
Yep.
That is case, and in fact, we just got an email this morning that another individual has been admitted to the NIH Clinical Center for possible exposure to Ebola, so they're going to be monitoring and treating that individual as appropriate. But I think it has not gone from one person to another within the U.S. I think that is correct.
So what's, to me, first of all, I'm very excited that the world is really paying attention. I wish we had done the same thing with HIV when it started to break in the 80s and 90s, but so that's encouraging, certainly, and that, for, if you will, first world care seems to be the answer, in addition to the kinds of diagnostic tools and immunity building tools and so on that we're talking about. But to me, that all reflects that there is a opportunities, let's say, to really build up the healthcare system globally. And the cultural issues, I think we've certainly all heard stories of communities that are afraid to let people in. I know that there was some talk in some countries that the government was trying to scare us into saying that Ebola happens but it doesn't really exist, and so on. And I don't know that there's an answer, but I think it's worth discussing in this context and hearing your thoughts on that delicate balance of bringing in the scientific method and scientific technology and trying to deploy quickly in a situation where there's distrust and lack of infrastructure and so on. There isn't really a question there, but I'd like to hear your thoughts on that part of this moment. Why don't you start, Dan?
Yeah, so I will.
Well,
Go ahead, Howard.
I think you have to build trust with the local leaders. So every village leader, every community activist has to be educated properly. They have to take the time to sit down and discuss with them, both understanding how the virus is working to kill their friends and relatives and neighbors, and give them an understanding of the process, and then explain what you want to do to help them fight this disease. I think without building public trust and knowledge, you're going to be constantly facing this. I mean, we see that in even polio vaccinations, that some places in the world consider the people administering the polio vaccines as a threat to their community, and they're killing them. So I think, again, you have to work at the grassroots population. You need the community activists to first be educated about the disease and then be educated about how countries are trying to help them combat the disease and help their infected patients recover.
Yeah, so I think Howard's exactly right there. I think what we need to do, and we did this after H1N1, which was lessons learned from a virus. And I think, Ebola is going to be the same way, but it's going to be more that social interaction as well. And I'm going to say this right out, scientists usually aren't good at that. I mean, we speak science, and we need those people that can, speak to the heart, and we need, people to be able to communicate at those local levels and understand their traditions. I mean, when they take and they bathe a person after they've died and wash away their sins, well, you still have to be able to do that, and you have to be able to do that in a way that's safe for them as well, because that's their long ingrained tradition, and we're just coming, and forcing it down their throats. I think we have to build that trust. It takes a lot to do these. I see a lot of organizations that are doing this, USAID, Clinton Foundation, Gates Foundation, a lot of those organizations out there. We need to do that and continue to build that trust on a global basis, because people tend to sit there, oh, it's over in Africa, it doesn't affect us. Well, we know it affects us, and it has this time as well. And so we have to start doing that. These are one of those lessons learned from Ebola, and we have to grow from this and get better at what we do.
An extension of that, a common denominator around the world are cell phones. And even in the smallest villages in Africa, people have cell phones. So I'd be curious if anyone in the audience knows of apps being developed to help educate people in their languages about infection like Ebola and about steps that can be taken to help combat it. Because that would seem to me as a common denominator that is accessible by a large segment of the population. And I don't know that it's being exploited in that way.
Well, it suggests to me an opportunity for some really unique collaborations between scientists, technologists, anthropologists, artists, and so on to communicate effectively. That's probably the way I can, we can find those kinds of teams and cultivate them. That may be the fastest way to get there while the technology is improving. We have another question from the audience. Does Thermo Fisher Scientific, let's see, do your assays support swipe tests to check surfaces and equipment for Ebola residue? That is, wipe down a surface, extract materials, then check for presence. Good question.
It is a good question. I mean, as long as the virus is present there, it will pick those viruses up even off of a surface. So yes, you could, but and fluids, bodily fluids is where it's usually contained in, so you would be able to pick it up from there.
Dan, do you have a specific kit for that, though? I think one of the questions is, do you have a swipe kit that could check different surfaces for Ebola?
Yeah, so our system is, a research use only system, so it's not an approved, doesn't have emergency use authorization. And the kit itself, it starts with, input. So you put the input into a Trizol or a MagMax, so you can do input from any source, whether it's a swipe or anything else. So yes, the short answer is yes and no. It's not specific for that, but it'll allow that to occur.
And the connected question is there what decontamination are you finding that's really worked? There's sort of two questions: what decontamination are you finding that's working the best, and what personal protection is working the best?
I wouldn't say I'm the expert at that. I mean, you get the moon suits that they're wearing that keep the protection there, and I would love to say bleach because that's the kill-all, cure-all that we tend to use, but I don't know what the recommended protocol is there. Howard, do you know on that?
Well, for the samples, Trizol is clearly inactivated. In fact, before I could get Ebola samples from my Army colleagues, they had to prove that the Trizol treatment of the blood inactivated the virus. So Trizol is clearly a method to inactivate the virus for then processing samples for research purposes, and even for the initial analysis, the initial extraction from the patient's blood, they're using Trizol. But it's not the perfect reagent, on the other hand, although I heard some talk the other day about some people were using a different reagent to get better quality RNA, but there was some concern that it was not, in fact, completely inactivating. But we can say for sure that any samples that you want to process for research purposes, Trizol does inactivate the virus.
Yeah, I think the question was more about decontamination, and you're right, Trizol is what we recommend and what the Department of Defense recommends. The CDC recommends MagMax. It does 96 wells, and it inactivates as well. But in terms of surface, I think that's a different question.
Well, this has really been very, very informative, and thank you so much, both of you. We have just a few more minutes, and I just want to give you a chance, each of you, if you have any final comments or anything that hasn't been addressed that you think we should mention in today's session.
Well, I would just like to add that I think we all have to work together. So for those of you on the line, if you're working on some agents that you might have ideas, don't forget the immune system. It's one thing to block a virus in a tissue culture dish. It's another thing when you treat the whole system. So get people who could help you with consideration of the immune system. But look outside. Don't be in a room with just mirrors. Be in a room with clear windows where you can look at others and try to work together with other groups to accomplish the ultimate goal of helping contain and treat this deadly virus and the viruses of the future, because this will likely not be the only outbreak of Ebola, but of other viruses, as there are indeed new and emerging diseases being identified on a sadly regular basis. So I would urge everybody to work together and reach out to people you need help in and get the help you need, but don't be silos. And so that's what I would add at this time.
Yeah, and I would echo what Howard says. We have to work together and look to different sources and look to different people. And then I'd also like to, again, go back to thanking, all the people that are out there. They're out in the front lines that are doing the work directly with patients. Those people are doing the research as well, because it's, it's a thankless job. Maybe you get a few publications out of something, but, really the work that you're doing is tremendous. So I want to, applaud all you for all the work that you do.
Well, thank you so much to you, to everyone who has been involved today. This is the website for the Biopharma Research Council. We will be coming back to the topic of global collaboration to address Ebola throughout the year, probably 3 or 4 times. So feel free to be in touch with us. You can find my email right on the website. You'll see the recording from today, and the slides will be posted probably about tomorrow, and we'll send a notice out on that. This is exactly, and thank you, Howard, for the note that we really have to work together. That we feel that what we can provide at the Biopharma Research Council is opportunities for people from different parts of the community to just learn about what each other is doing and potentially to stimulate fresh collaborations. And this has been a very great example of how that can work. So thank you so much. And with that, we'll say goodbye and see everyone in 2015. Thanks so much.